All right, well, good afternoon, and thank you for joining us. It's my pleasure to welcome you today to our interactive program with Associate Professor Shai Shen-Orr. Today marks Professor Shen-Orr's return to the West Coast, and while we wish it could be in person, we're delighted to have him here in this intimate virtual setting. For those of you who don't know me, I'm Heather Dula, board member of ATS Southern California. My husband and I became involved with the Technion over 8 years ago. We both work in healthcare and clinical development, and we felt that Technion represented a lot of areas of interest to us, from advances to medical research to Israel's defense. It was something we could both be involved in. And then today, in addition to our Southern California community, I'm delighted to welcome friends from Seattle, San Diego, Scottsdale. We live in different cities and states, but we all share a passion for Technion. Let's see, today Shai will update us on how his pioneering research is unraveling the complexities of the immune system and unlocking the potential for personalized medical treatments. Addressing human health is one of the greatest challenges of the 21st century, and the Technion is undertaking one of the boldest and most ambitious endeavors since its inception. One that will be unmatched in Israel with the creation of the Human Health Institute. The Institute will break down silos and integrate faculties such as engineering, medicine, and computer science to encourage a multidisciplinary approach to teaching, research, and clinical practices, and foster greater collaboration with Technion-affiliated hospitals and industry. Associate Professor Shai Shen-Orr heads the Multidisciplinary Systems Immunology and Precision Medicine Laboratory at Technion Rappaport Faculty of Medicine. Born in Israel, he's lived in the United States for several years and is a third-generation Technion alumnus. He earned his bachelor degree at Technion, his master's degree at the Weizmann Institute of Science, and his doctorate at Harvard University. He completed postdoctoral studies at Stanford University. Very impressive, I have to say. And just a few housekeeping rules before we begin. Please make sure that you mute yourself during the presentation. We also suggest that you watch this program in speaker view for the best experience. And then after the presentation, I'm going to ask a few questions, and then if you'd like to ask a question at that point, you can literally just raise your hand, or you can, we'll call on you, you can send a question in the chat, and then we'll go ahead and we'll unmute you, or you can unmute yourself. So now it's my pleasure to welcome Professor Shen-Orr. Take it away.
Thank you, Heather. Can you, everybody hear me? It's a real pleasure to come back to the West Coast. I toured with ATS and visited with Pamela. I guess it was, well, sounds like, feels like many moons ago, but I guess it was a year and a half ago, I think, or something like that. Wow, life has changed. So let me start. I'll just share my screen, and hopefully people will see the presentation. Whoops. Okay, okay, people see my screen, I hope. All right. So it's a real pleasure to talk to you today on our research. As Heather said, I lead quite a diverse lab that kind of really combines healthcare, kind of science of biomedical research for immune-based precision medicine to kind of more basic questions in biology on how the immune system, kind of what drives immune variation and so forth. And all that really heavily rooted in computational analyses, algorithm development, and so forth to really make sense of this data. And what I hope to achieve in the short time we talk today is to describe to you really where are we going with this ability to monitor the immune system and what this will allow us to do. And this is kind of work that we've been really tackling in the lab from multiple fronts. And the driving force for this work is really a very simple question that I imagine can happen, and one where a patient goes to a doctor's office and just asks, "Hey doc, how's my immune system doing?" It sounds like a trivial question. You'd hope that there's a good answer for this, but really, in fact, there isn't one. And the physician would if push comes to shove, the physician will have to admit they don't really understand one's immune system. We don't understand the variation within individuals, how we vary over time, and certainly between us. And I think we see this unfortunately quite quite acutely right now with the COVID pandemic. Now, just to kind of give you a frame of reference to how ridiculous this situation is, think about what if the patient had asked, "How's my heart doing?" Right. So if he'd asked that, the doctor has very accurate measures to actually assess cardiovascular health. They have recommendations for lifestyle changes. They have recommendations. They can go to different treatment options, from very interventional surgeries to therapeutics to prevent deterioration. With the immune system, that's not really the case. And in fact, it's that gets to a problem, because this is a system that's really cardinal to medicine, to all of medicine. Basically, every disease, if you're talking about infectious diseases, cancers, treatments such as vaccination or autoimmune diseases, basically the immune system is critical in almost all diseases. And we don't really understand this immune variation, and particularly we don't understand maybe the most acute places that it varies with age dramatically. In healthy individuals, our immune system just changes dramatically. So basically, if you look at anything that we measure on this axis, on the y-axis here, and you look at young people and old people are much more variable. They're all over the map, and no one really understands what drives this variation in the immune system. And this is critical because of the implication of the immune system for clinical health, and it's clear that that variation could play a role. Now, if we go back to this patient's question to the physician, and we try to understand what's going on, well, basically, this is a system that's all about sensing the environment. It's the internal environment or the external one, and adapting to it. This is basically a sixth sense, right? It continuously senses what we're seeing and what it sees, and it changes to protect us. And as it does so, it generates more and more variation, and that variation actually then plays a role in clinical effects that we see. And particularly, this has been acute in any kind of immune-mediated diseases and immunotherapies that are kind of the leading treatments today. So if I go back to what the system's all about, basically this is a cellular system. The quanta unit, the atomic unit of the immune system, are cells. And this is I draw here one cell, but really the immune system is about many different cells that are interacting, and we've been able to assess them, and we do so. And I'm sure everyone here on the call has done so. When you go to your HMO and you get a physician's, you get a blood test, and you get, within the blood test, one of the most common tests done is a complete blood count, and within that is the white blood count. And you basically can describe from your blood the 5 different immune cells that describe your entire system: neutrophils, basophils, eosinophils, monocytes, and lymphocytes. And they have these parentheses, and they have a range, and you can look at the variation, and the physician looks at it. But even if you see just this little plot here on the right, you basically see that there's a lot of variation. Every yellow point is one person here, and there's a lot of variation just in one cell, the lymphocytes, and that changes dramatically with age. And this test that we all do has been around since 1957. Okay, so basically we've been testing these 5 cells since 1957. The only real change has been that the test has become automated, and one may wonder if anything's changed. Well, what's really changed is that we now understand the immune system in a much kind of better resolution. And we understand that when we look at lymphocytes, we're actually talking about hundreds of different types of cells, each of which is different and has a different function. Each has a different, a different component that it provides to the immune system. And you can sort of think about this like we needed to quantify all these different cells. But for many years, this was just not possible. We couldn't really capture all of these different cells. And in fact, even today, we measure them with this complete blood count test that I showed you. So all these tests together, all these different cells together interact, and what they yield is what we call an immune response, which is what yields our protection. It's basically an emergent phenomena of the interactions of these different cells that do so. And what's changed in recent years, and this has been basically for the past decade, is that we now have technologies that let us, from a single blood drop or a biopsy, get the entire system. Right, so I hope you can see this on my screen, but basically this is Mona Lisa from coffee cups, and we can get this kind of emergent phenomena that we couldn't get before. And when we look at your immune system, we can basically do the equivalent of what I just did as we shifted these two slides. And just to kind of give you the equivalent of this white blood count that I showed you that's been around for basically close to 70 years, this is what I would call a modern day capability of doing so. This is a machine we have at the Technion called a CytoReason, and you can give it, you basically can take a blood draw and on it measure basically 2 to the power of 40 different types of cells. And what you see on the right in this cartoon, and I won't go into the details of it, is basically a modern day blood count, if you like. Every circle here is a different type of cell we know has a different function, and we can quantify all of them together in a person, where the size of the circle tells us how much I have of each one of those different cells, right? So we can really basically quantify what your immune system looks like in every given point in time, just which was basically science fiction up until a few years ago. Now this capability now can be applied to make a dramatic shift, and that is basically to move immunology and understanding immunology from a field that's basically been all about studying mice in cages. They're obviously inbred animals. They don't live in the real world. They don't live for the same number of years as we do. They have, they don't really have the same immune system, but they were easy to study, to a point of time where instead of studying the mouse and trying to understand humans, we can basically study humans in the real world, understand what their diseases look like, and basically use this information to try and directly kind of accelerate our ability to treat or diagnose different diseases. So this is basically a capability that was unimaginable up until a few years ago. And some people call this reverse immunology, because for so many years we were just moving from the mouse to humans, and the failure rate between mice results to actually curing humans is just from a point where I have a human drug that I saw in mouse to actually succeeding to cure a human, 1 in 10 succeed. But if I start from a mouse result at the beginning of an experiment, we're talking about basically 95% failure to actually cure humans in the end. So being able to move the other way around, start in humans and actually understand what's going on, if need be, actually do small amount of experiments that are needed in mouse, is a dramatic change for our ability to kind of accelerate healthcare. So this is basically what we study in the lab. We study humans, the human immune system, to actually understand how it works and how we could develop immune-based precision medicine from it. Just to kind of show you this one slide describing the 2 different approaches my lab employs. One is on the left-hand side, we perform very focused, high-resolution studies where we take samples from individuals, either blood or biopsies, and measure basically everything we can in them on the book. So we switch from measuring, what up until a few years ago was, I guess, 10, 20 different things we measured, to getting on one person hundreds of thousands of different measurements of their genome, their genes, all their different cells, what the cells are doing, and the proteins, how the cells are communicating. All this information we're collecting from every sample, and we try to understand the disease. And simultaneously, we leverage all the amount of the data that's actually now available on the internet in the public domain. So basically, since 2005, if people want to publish a paper, they used to be able to publish the paper without providing any of the data. That's no longer the case. Basically, for the past 15 years, we have to deposit the data on the internet, and that's really accelerated our ability to do research, because we can just now, basically a high school student can download the information and start studying human disease and make discoveries. And we take these two approaches and they complete one another, because one of them is focused and expensive, but gives a lot of information, and the other one doesn't give that same level of resolution, but gives us a breadth of ability to look at many different diseases together, and doesn't really cost a lot. So this is kind of how we do this, and the students in my lab are very come from a very diverse background. They can start from computational computer science and pick up the biology, or they start from the biology and pick up the computer science. We embrace diversity. Just to give you a sense of the type of research that we do. So here's one story. Actually, we've just now submitting its follow-up on the type of information we can get at. This involves looking at inflammatory bowel disease patients. So IBD, Crohn's, and UC are the two diseases. Second-line treatments in these diseases are what's called immunotherapies, and particularly anti-TNFs. These are basically one of the top-selling drugs right now in pharma, and unfortunately, basically on the order of 30% of people respond to these drugs, and it takes a physician today 16 weeks, so basically slightly over 3 months, 3 to 4 months, to actually decide whether the patient that they gave the drug to will respond to it. And what we're able to do is take these individuals, develop very tailored algorithms, or we take measurements from colon of inflammatory bowel disease patients before they get the treatment. And from this, we were able to identify biomarkers, particularly immune cells in the gut, that allow us, before they get the treatment, to know whether they respond or not. And moreover, we were able to take this and again develop a whole suite of computational algorithms that allow us to then take the information from the gut and predict what it looks like in blood, and show on an external validation cohort that we can predict from a blood draw before people get the treatment whether they'll respond or not to the treatment. And so this is kind of the direction that we've been moving forward with IBD, and we continue doing this for additional drugs in inflammatory bowel disease, where we can now take a blood draw and start directing a patient to the right therapy without needing to do these kind of trial by error experiments. So that's one direction. I'll take you to the other one, which we're really excited about, and this is the idea to really answer that patient's question on how's my immune system doing. We basically need a metric, right? We want a metric that defines immune health. So think about if we took all the people on this Zoom and we wanted to try and understand who has a good or bad immune system. That's not a really trivial thing, as opposed to if we organized ourselves by height. That would be easy, right? So, but as soon as we talk about the immune system, that's not really trivial to do. And what we've been doing over the past over 10 years now is following patients over time. This is a study actually started at Stanford, and we collaborate with folks at Stanford for this, Mark Davis in particular. And we published a paper, this is, I guess, now 2 years ago or a year and a half ago, where we followed patients for 9 years, and we looked at how their immune system changes over time. Now, I mentioned that the doctors don't understand, everybody looks different, they don't understand the variation. Basically, what we discovered is that when you look at patients over time for a very long time, and not looking at the complete blood count, white blood count I showed you, but at a high resolution that we can do now, what we identified is basically that people change along a very conserved pattern, right? And you can see on the right-hand side here a graph showing you people of different ages, from 60-year-olds up, and a cell called the naive CD8 T cell. We know that this cell drops with age. You can see the black line showing you that on average it drops, but every colored line here is one person, and they look like they're changing at very different rates. They don't change like the average. And what we realize is that basically there's a fixed pattern that everybody changes, and the only thing that matters is that's really different is that they change in different rates. So just to kind of put this on a less kind of scientific graph, imagine basically what I'm trying to say is that if I looked at your immune system at any point in time, we could predict what your immune system is going to look like. And the way we would do this is basically by this identifying that we all change along one road. Okay, so we're all starting from point A on the left-hand side and we're driving to point B on the right-hand side. And the only difference between us is that we're driving at very different rates. Pam may be driving a Ferrari and I'm driving a scooter, right? And so it's very different rates. We look very different because it's changing very differently. We're affected by the environment, but we're able to actually predict where we are on the road. And by predicting where we are on the road, we can predict what the clinical impact of this. We call this position on the road the image score, right? The position along your immune aging trajectory. And what we've been showing repeatedly is that this actually gives us clinical utility beyond what's captured today in any kind of clinical lab tests. And so just to kind of give you where we're going with this, basically with this metric, the image metric allows us to start making sense of blood. We're basically developing a diagnostic kit for immune-based precision medicine. We're moving from the left-hand kind of CBC, the complete blood count that's been around since 1957, to a very high-resolution assessment. A high-resolution assessment of your immune system. But we're doing this we're now developing it to be cheap, where we're measuring different immune cells in your blood simultaneously. We understand now much better what the norms should be, that we can make them much more tailored to your environment and age. And we study the interactions between them. And we already have some exciting preliminary results where we can show that the immune aging metric, the image metric, it shows significant differences between HIV patients and healthy controls that are age matched. Patients going through trauma have shifts in their immune age, and we're looking to see what the clinical implication of this is. When you get an infection, your immune age changes and then drops back. What's really exciting is that we've been able to find in blood, detect a strong correlation between the immune age metric and cognitive decline and all the way to Alzheimer's. We're seeing this change in people that respond to immunotherapies. And most interestingly, perhaps in terms of understanding our environment, is that children in the developing world seem to have a much higher immune age than children, the age-matched children in say Palo Alto. And so we're starting to kind of understand what is it that drives these kind of shift, the rates at which you drive on the road. And from this, we hope to develop better therapies and tailored vaccines that match your immune aging. So I don't know how long I still have. Do I have a few more minutes? I hope. I'll just kind of, I hope I kind of give you a sense of what my lab does. And I'm sure what you felt is that, wow, there's a lot of data that's happening there. And in fact, this is true. Data has been growing like crazy. This has been really the case since the mid-90s, not just in biology, but all over in many different fields. And the problem has just been that the insight has also been growing, and we've been feeling great, but the gap between what we measure and what we understand has also been growing, right? So we're just not getting the bang for the buck for all the data that we're generating. And so this is another thing that my lab works on, is how do we make more sense of the data? How do we reclaim the biology from all the data that we measure so that we can just accelerate our research further? And to kind of give you a sense of how we do this, the real answer is that we build models that represent, or simulations that represent the tissues, but to give you a sense of how this has kind of where this has happened and already changed your life, I think has been the phone industry. So I imagine many of you remember, we used to have these phones, they didn't have any data. Life was simple. And then came the Nokias and they collected data, and we had a GPS, and we can kind of browse these phones. And so, there was big data, everybody had a phone, but really nobody was doing anything with the data that was being generated. It was just kind of uploaded to the phone provider, and aside from billing us, nothing was happening. And what changed is that we took all this data and we started modeling the real world. And so now basically the GPS is used to actually model the streets in our city, model the traffic, and suddenly our reality is augmented in a way that totally transforms our lives, right? And if I forgot my Nokia at home, it wasn't a big deal. I probably kept going to work. If I forget my smartphone at home now, I go pick it up again because I can't really imagine going through a day without this augmented reality. And so similarly, this is what we've been building in biology. So I co-founded a company called CytoReason, the chief scientist of this company, and basically CytoReason has been developing through algorithms that we originally developed at the Technion, the biology of the future, a shift from this area where we are not keeping data, and in some places in biology is like the analog phones, we still don't store data, to all this kind of data that's being collected since the 90s, to actually modeling what disease looks like so we can transform healthcare in a much faster fashion. And just to kind of give you the shift that this describes, we're moving to a place where instead of just looking at the data, we're modeling disease in a way that takes all the data and all the knowledge, all the literature that gets published. Every 30 minutes, a new paper gets published these days. And instead of we're modeling from all this information, we kind of encapsulate it and capture what the cells are doing and what the, what are the cells in the tissue? What are the regulatory programs? How are the cells interacting in networks? How the disease changes over time? How do individuals, how this relates to the clinic? All this information is now put in a simulation to actually really accelerate drug development and healthcare. So that when your physician looks at your blood sample, he's able to actually give you much more informed information about your immune system's health and how it relates to treatment options. And so that's where we've been heading. Just to put one slide to shift, we're basically moving from this too much information kind of phase that we've been in for, I think, the past 20 years. More and more data comes our way, more and more information. You can figure it out if you're an expert in the field in a particular room, it kind of feels okay. But as soon as the data encompasses all of the immune system, all of the medicine, and we need to start making sense, we need computational models that allow us to naturally organize the system and just get higher utility. So with this, I'll end, and I hope this was...
It's great. Shai, thank you so much. Your work's incredible, so relevant.
I have a question.
We appreciate it. Before we open it up to Q&A to the audience, I have a few questions on my own. The first one is not clinical. What inspired you to go into this particular area of research? Any life event?
Yes. So I actually, I literally stumbled on biology by accident. I went on Technion sent me on a student exchange program to UPenn. And from this, basically, I just fell in, I needed a part-time job to finance my time. And UPenn Biomedical paid $15 an hour, which was twice student wage. So I decided to take the job. And that was awesome. Suddenly I felt like I was doing not just cutting edge computer science, which was what I was kind of was studying at the Technion, but suddenly adapting it to healthcare. And as so I went from studying, I went back to Israel and I started studying at the Weizmann how bacteria behave and how I can use computers to actually make sense of what happens in bacteria. I said, that's really great, but the taxpayer really isn't getting what, maximizing on the luxury of me studying bacteria. And so I said, well, I'll shift. And I shifted to studying a little worm that's 1,000 cells big. And that's what I did for my PhD at Harvard. And as I did so, two things happened. One was that the human genome was sequenced, and suddenly there's more data to study humans. And so there's a realization that we can, instead of studying the worm, we can do it in humans. And at the same time, my father was diagnosed with lymphoma, and I was getting his clinical tests, and I had more information about the worm than I had about my father's immune system. And it just seemed to me ridiculous that that's the case. It was clear to me that it could change. It just wasn't happening fast enough. And so I finished my PhD and decided to really kind of, this is 2007, I decided to study the human immune system at a system level. It was unheard of at the time. It seemed crazy. So that's kind of, life takes us in this directions.
Thank you. Question. So the mass cytometry that you discussed with the labs and being able to look at all these different aspects of your labs now, is that something that's available yet at a lab like a Quest or a LabCorp, or are they still just looking at your basic labs?
Yeah, not yet, not yet. So just to give you a sense, this new technology that I measured, the mass cytometry or the CyTOF, that measurement per person costs on the order of, like, when we do it in the lab, takes us about 2 days to prepare and costs about $250 per sample, and we can do 20 a day, right? So not huge, but what we're doing now is taking this image metric and the technologies and bringing it down so that it costs a few dollars and can be done on a large scale at your HMO, at Quest, for thousands of people a day.
And so how far off is that, would you say?
So from a technology perspective, we're at a point now where I'd say probably a year off. From a regulatory perspective, it's going to take a bit longer, right? So we're now starting a lot of different, I'm busy writing grants, trying to basically take this to the next step, show, this is a diagnostic, the kind of accelerate the regulatory process for it. And I imagine this is probably, on the order of 5 years, I hope, away from.
Okay.
So
that's a ways off. This probably leads to my next question. I probably just answered for myself based on that answer. In terms of what you said, we're working backwards now with clinical trials. So you might not start with a mouse, but you might be able to take the human and work backwards. How would that impact the clinical trial process and preclinical trials?
Yeah, so basically what we're seeing already, so the company I founded, CytoReason, works with the leading pharma companies in the world, Pfizer, GSK, Janssen, and so forth. And what we're seeing is that it's accelerating the process, right? So, and it's decreasing the failure rate. So instead of studying, starting in the mouse and then finding out that it won't work in humans, already at very early stages, we're kind of inputting this human data simulations, and seeing how the next generation drugs are behaving. And so we're starting to see, just to give numbers, if you look over the past 10 years where computational biology and precision medicine have gone into drug development, basically the success of drugs has quadrupled. So drugs that have where we understand the biology earlier on with human data have basically 4 times the chance of succeeding, and that's going to decrease cost of development, and therefore really bring an age of precision medicine where it's worthwhile to develop a drug for a few people, which is currently very costly.
All right, thank you. So let's see. Jose, did you want to look at the chat for the questions we've got come up?
Yeah, I think Bruce had a question. So
here we go.
Unmute yourself.
Yeah. Well, what you've done and what you're doing is really interesting.
Thank you.
Initial question was, how do I, as a human being or as a body, I'll call myself a human being, whether I am or not is certainly open to question, because we are a complex organism. I don't think there's much question or much doubt that what we bring to the table affects everything that we eat, sleep, drink, whatever, anything that we put into our body. And it gets put into a system. I'm going to use myself as an example, that's somatic. So if I came to you and I have to do all this work, how do you begin to even think about dealing with the human factor, okay? And who we are as a person in a species, okay? that has the kinds of issues and problems that we have living in the world that we've created around us, okay? So that my body, my mind, my spirit can be involved in the process of doing whatever needs to be done in order to make my immune system work better. That requires not just what you give me in terms of ingesting something, okay? But it really depends on what table am I in? How do I evolve myself to where I can take advantage of everything that you learn about this, because it's all individualistic. I mean, of course, there are patterns and stuff like that we all fit into. And ultimately, it's not like you can give me a prescription for how I think, how I walk, how I live, etc., for making a change internally so that whatever's happening externally can be involved in the process. That's my question.
Sure. So it's a wonderful question, and the answer, I guess I'll try and
I don't
simplify.
know.
So I think that, well, it's not, I don't know. It's basically a transition. It's a transition from, so if you look where medicine is, or, to a large extent, I'd say is shifting, was a medicine of averages, right? So on average, a drug helps people. Now, when like any new technology comes in, there's a lot of hype, and the idea of precision medicine, those ideas were not called precision medicine. If you go back 10 years, people were saying personalized medicine. Okay, and the difference is was, when it first started, it was like, okay, well, we're going to be able, now we can measure basically anything on humans. We'd be able to really get to exactly what a person needs, and that is, we're not there yet. But we are in the middle. Those new technology, those new computational algorithms we've been developing, and others are basically taking us to a point where we can subgroup diseases and principal components, or kind of chief factors that are driving your immune health or any disease, to a point where we are more precise. And by being more precise, we can give you more tailored advice. We're still, we'd still struggle with saying exactly what's right to you. We're making those attempts. We're getting better all the time. But already what we've done is science fiction compared to where we were a few years back. And in fact, I think what's really promising, and you can kind of see this in the graph I showed of the data going up, is that the rate in which we do this, and maybe actually the biggest example is the vaccine development for COVID, is growing exponentially. We are improving in our ability to learn from data and to improve and give you better healthcare and more tailored treatments in an exponential rate compared to what it was. I mean, just the idea of being able to develop a vaccine in a year is, go back 2 years, no one would believe that it's possible. And the vaccine went through all the right regulatory approvals and everything, right? And so there's a lot of this going on in every facet of healthcare. So it's a new era, but yeah, we're not yet at a place where how we exactly, by understanding your thinking, we know how to recommend. Though I have to say, maybe it's a good place to just mention, we did a study looking at placebo responses and how they affect the immune system. This was actually done in mice, but what we were able to show was basically that if we stimulate the area in the brain that is associated with placebo response in humans, we get activation of the immune system, we get stronger immune response, better killing, better memory responses. So even your thinking changes your immune system.
Janie, do you have a question, Janie?
Well, this is all fascinating, absolutely fascinating. And I am so impressed with what you're doing, Shai. I can't wait to come and visit you in your lab and see firsthand what's going on. So my question to you is, there is so much going on and it's so wonderful. What aspect of this just excites you the most about being connected to this institute?
So I think this Heather mentioned this early on. The initiative that's been going on at the Technion just kind of recently, the Human Health Institute,
Mhm.
is probably the thing that excites me the most now. So as a lab, I've been a part of the Technion. Technion's a great institute, but basically my lab, largely has been working on its own. Yeah, I collaborate every once in a while with people, but what we do is driven just by our capabilities, and what the Technion has put forth on the charter with the new president that we have is basically no. Let's take the strength of the Technion and make it work together. And particularly, they outlined human health as the first initiative where that's happening, and already that's totally changing the map. So we are studying human aging now, but rather than it just being focused on my lab, or kind of what the stuff I showed you in the immune system, we're now tailoring this with people in biotechnology that are doing protein kind of food design for the elderly, so that we can check how we can tailor diets and we can tailor specific foods that would alter your immune system. We're doing this with people who are studying in cells how your metabolism is affecting immune cell responses, with clinicians from Rambam Healthcare across the street from the Faculty of Medicine that are giving in the integrative view, with people studying cognitive neuroscience and how that relates to motor behavior, and all of this on the same person, bringing in all this information together. And I think this is really can only happen in an institute like the Technion, and there are very few where medicine and top-notch engineering sit together. And so it's just been really exciting to be part of this.
It sounds fabulous. And I wish you all the best. And I can't wait to be there in person.
Thank you. Likewise. And I'd love to have you over. Everybody here on the call, more than welcome.
Well,
as soon as you can fly.
I'm co-chairing a mission for the Technion for entrepreneurial women and women who are interested in science. And we're hoping to be there right around board of directors time in 2022. So look forward to seeing you
See
then.
you soon.
Yes.
Yes. And Nathan, I think you might have had a question, or I can ask it on your behalf. But if you want to unmute.
I can also unmute myself. Hi, Shai. Thank you very much. It was a great presentation, as always. And so I just wanted to ask, you started out talking about inflammatory bowel disease, and do your sub-classifications of immune cells currently help you to tailor specific medicines to specific patients, or is that still too early?
So again, so from a results perspective, we can now, and we validated this in several external cohorts, we can predict infliximab response to anti-TNF. In fact, what we've recently shown is that we can do this both in inflammatory bowel diseases and rheumatoid arthritis. So on a blood draw, we can say ahead of time whether you'll respond or not. Currently takes over 3.5 months. We can do this for vedolizumab, another drug, and we're continuing with others. We're looking to see when you lose response to a drug as well. So there are some people who respond to a drug and then lose. What we're envisioning is a blood-based test we're just started with IBD for a variety of scientific reasons, but I think the future holds that for many diseases where where we can make sense of blood, and so that the physician response, rather than right now being dictated by the physician's kind of guessing kind of basically the payer telling them, this drug costs more or less, that the information would go more and more tailored. And we could basically say, look, you're not going to respond to this drug. We're going to take the drug that would have been third in line. You're getting it first because, our blood data would tell us that, 85% chance to respond to it versus the other one. So that's where we're heading. And so right now we have basically 2 drugs done, and we're continuing with the few others.
Okay, but so currently you cannot tell a physician, still guide him. You can tell him what is the likelihood that you're going to respond, let's say, to an anti-TNF and so, but you couldn't tell him use an anti-TNF versus a JAK inhibitor in this particular patient.
No, we can. I mean, the question is, from a discovery perspective, yes, we can. We can go to the physician and tell him, you should use this. Whether is it already in the kind of pair guidelines to change the decision and so forth? No, not yet. We're working very hard to actually, including with Anthem in the US, to start taking our diagnosis to a point where it's actually affecting pairs' decisions so that they prioritize properly the drugs. But there's always, as Heather has before, from the scientific discovery to actually making this from a commercial viable sense and convincing the regulatory approvals, not everything gets the same priorities as COVID vaccines do. Let's put it this way.
I mean, in Israel, you have beautiful cupatrolims, and so that actually is probably, it's easier probably to get it done in Israel than in the US. And so any traction there in introducing those types of tests for those patients?
So we, you're right, and we have ongoing collaborations both with Maccabi and Clalit, and to do so, we're not there at a point where it's being recommended yet, but I think we're close to there. I expect, I hope in the next 2 years we'll start being able to drive decisions in IBD in Israel on this.
Thank you so much. Thank you.
Victoria?
Hi, Shai. It's great to see you. I had a question for you about COVID research. I'm wondering if you're going to, if you have the access to and you anticipate being able to work with data on patients who have had COVID and recovered to see what it's done to their immune system.
Yeah, so wonderful question. Hi, Vicky. Yes, we have, so basically there's 2 projects we've been doing in the lab, or maybe 2.5. So one, we've been looking at the relationship of immune age to COVID severity, still early days. We've been studying the COVID vaccines, the Pfizer vaccine, and basically this is the world's first mRNA vaccines, and we have some really exciting results there that are ongoing. Seems like quite potent. And then from directly the question you've asked, with Lior Gepstein, who heads cardiovascular medicine at Rambam and is also a faculty member in medicine, we're starting to look at post-COVID kind of side effects, particularly cardiovascular side effects, and trying to understand what is it that drives them. And so this is a collaboration that just recently started.
Okay, thank you.
Thank you.
Doug has a question. Doug, did you want to unmute and ask your question?
Okay, so first of all, can you hear me?
Yes.
Okay, great. Shai, through our crowd, I invested in CytoReason. I am a total believer in the work that's being done. I happen to live with 2 autoimmunes, and I have a kind of a 2-part question. What other autoimmune issues is CytoReason actually pursuing right now, or Technion's collaboration with CytoReason? Are you doing more of a broad-based research, or is it very specific to certain autoimmunes?
Right. So first of all, thank you for investing in CytoReason, Doug. And I am a true believer as well, but need funding. So
I hope so.
Yes. So basically, CytoReason has been working with pharma companies, and so what we've been, so in many ways, as a startup, it's right now 45 people, profitable, sits in Israeli and Tel Aviv, but is still dictating in terms of how we prioritize based on what interests pharma. So interestingly enough, if you look across drug companies, what interests them repeats, and so. We've been looking, if we're talking about autoimmune diseases, we've been looking inflammatory bowel diseases, both UC and Crohn's, RA, lupus have been places where we've kind of looked. Beyond that, we've been doing things in asthma, in psoriasis, atopic dermatitis, and oncology, immuno-oncology. So that dictates it. In the lab, my lab right now is basically focused on kind of 2 different directions. One is kind of developing the computational methods to represent these disease model simulations. So that's research that's driven kind of computer science students that are kind of picking up biology. And on the other hand, I have physicians and biologists who are kind of using these algorithms to study immune variation, and there where we're focused predominantly. So first of all, studying healthy aging. We obviously, this is a big world problem that we think we need to understand in this kind of question I posed, but also sepsis, which is also a problem in the old. So severe infections, not only is it a high mortality in the clinic, in the hospital, but post-release, percent of patients who've had sepsis that unfortunately pass away in the few months after releases in their tens of like on the order of about 20 to 30% of patients would pass away in the first year or so. And so we believe the immune system state in which they leave the hospital is a big component of this. And so we're looking at that. And inflammatory bowel diseases, where we are part of a nationwide consortium looking at IBD, and certainly there we've been in a leading position in the world. Israel has the largest repository of inflammatory bowel disease patients that have been phenotyped. And we're doing quite multidisciplinary work there across actually multiple Israeli universities, where we're synthesizing all this data into one model.
Thank you. I'm cheering for you.
Thank you.
Thank
Absolutely.
I'm doing a last kind of look of seeing, but I actually had one last question. I mean, it seems that a lot of our Technion alumni eventually return to the Technion to, be professors, do their research. So was that always your plan? And maybe what makes Technion so special that this was the place that you wanted to come back to?
Yes. So funny enough, so I never intended to be an academic, but it was always the plan. Once I figured out that I was going to be one, it was always the plan to go back to the Technion. So
You have the accidental career path from the start, right?
yeah. Yeah, I mean, I think, I think me and other faculty members, I mean, I think Technion is just a great institute to come back to. I mean, I have to say, I was offered, positions finishing my postdoc in the U.S. and leading U.S. universities. The Technion's ability to be competitive, and competitive, I mean not by the salary I get, I mean by the scientific equipment, the ability to actually give me a lab that can do cutting edge research was on par to what I would have gotten at Stanford. And that is obviously chief for anybody who thinks about doing science, is that they're going to be able to do the kind of most leading science. And then the environment is amazing. So the ability to interact multidisciplinary, if it's life sciences to engineering, is and just across engineering disciplines as well, is quite unique. Doesn't exist anywhere else. And to me, coming back to Israel, and this is a few years now, but basically realizing that on one hand, I wanted to do, I mean, I guess there on three different directions. I wanted to do precision medicine, so that's medicine. I think about the immune system and anything in biology as being basic research that one needs to understand. The immune system is a complex system and how it evolved and so forth. So you need that basic life sciences and basic research capability and the engineering side. And thinking about that, I just couldn't find another place that made sense. Now, also being born in Haifa and a third generation alumnus, who gives me an option even? So certainly not my mom, but no, I'm kidding. I wanted to come back.
Amazing. Well, thank you for sharing that. And I just want to thank you again so much for taking time out of your very busy schedule to be with us today. It's always wonderful to hear about your work, your personal story, your research, I mean, which is especially relevant today. And we really look forward to hearing updates about your work and your successes, and hope even more that we can be together in person in the not too distant future. You are always welcome on the West Coast, always welcome in LA and our cities on the West Coast. And for those who don't know me, I'm Pamela Wohl. I'm the ATS Senior Director of Development for Southern California. And I want to thank Heather for moderating and hosting our program, and thank all of you for being with us today. I really hope that today's program renewed your pride for the Technion, and I hope that we can see you at our future interactive exclusive programs for Southern California. Our next one up is April 5th at 7 p.m. It's our Cocktails and Conversation with Technion alumnus Elliot Peretz, who's with NASA. He has a fantastic, really interesting, inspiring story. And on April 28th at 10 a.m., we'll be featuring Professor Shulamit Levenberg. She's the Dean of Faculty of Medical Engineering. She is a powerhouse. She's a leader in tissue engineering, diabetes, regenerative medicine, 3D printed meat. Like, there's nothing she doesn't do. Hopefully you've seen the save the dates. You will be receiving the invitations. If you have any questions, please don't hesitate to reach out to me or Vicky in the Bay Area or Mark in San Diego, Phoenix, Palm Springs. And we hope that you enjoy the program and enjoy the rest of your day, and hope to see you soon. And Chag Sameach, happy early Passover. And thank you all.
Pamela, do you have a recording of this?
Yes.
It was so good. It was really, he's an great lecturer.
Thank you. Yeah.
Thank you. And thank you all for coming and supporting us.
כל הכבוד.
תודה.
כל כך הרבה מערכות הסברת.
Thank you.
Thank you.
Thank you.
How could I get it, Pamela? Can I?
Honey, I'll send it to you.
I'll
I'll send it to you.
Thanks.
Right.